it is my great pleasure to introduce our speaker, Professor Renaud Jolivet, who is a full professor at Maastricht Centre for Systems Biology, and also a chair of the Neuroengineering and Computational group at Maastricht University. He was nominated a representative for individual researchers and innovators at the European Commission's ERA Forum, the chair of the Science and Technology Committee of EBRAINS, the organization that we as the Institute joined recently in March this year, which is the organization for the European Research Infrastructure for Neurosciences, and holds also a courtesy of appointment at CERN in Geneva, the particle physics laboratory. He has previously served on boards of the Initiative for Science in Europe, Marie Curie Alumni Association, and Organization for Computational Neuroscience. He was also a 2023 Neurotech Fellow of the Foresight Institute, and he's worked in many countries, Australia, Switzerland, Japan, the Netherlands, and the UK. His work focuses on brain's heterocellularity and on neurotechnologies to interface with brain tissue. In 2023, he received both Marie Curie Alumni Association Career Award and the André Mischke Young Academy for Europe Prize for Science and Policy in 2023 for his achievements in science and in science policy. So I know the stage is yours. Thank you very much again for joining us, and we are very excited to hear your talk.
Thank you. Thank you for the kind invitation, and thank you for the introduction. And clearly I do way too many things, and so I'll say a few words about that at the end. So today, so I was asked, so I'm a neuroscientist and I do some science policy, so I will try to awkwardly connect these two things. So I will talk first about one question that I think is very important for today's neuroscience, and some work we've done towards trying to address that question. And then towards the end of the talk, I will talk a bit about my career in science policy and try to explain what it is about, why I do it, why I think it's important or not, and how you can maybe benefit from this from a career perspective. So at the back of the title here, what you can see is a 3D reconstruction from an artist of some of the cells at the cortical level of a rodent brain. This is from the Blue Brain project, or the defunct Blue Brain project in Lausanne, and you can maybe distinguish big yellow strands. These are neurons, and there's also vasculature in red, and also some other cells there, which are astrocytes, which, if you're a neuroscientist, all of you will be familiar, are a subclass of glial cells. And in fact, this idea of interaction between neurons and glial cells is, I think, one of the grand unresolved questions for modern day neuroscience. And so why I think these glial cells are very important? I mean, one reason, which is not a very good one, obviously, is that there's a lot of them. So this is a paper of computational anatomy paper from Azevedo and colleagues. They looked at human brains. You can see in the top left corner that your brain contains about 170 billion cells, half of which are neurons. But in fact, if you focus a bit on the numbers, you will see that most of these neurons are contained in the cerebellum. A lot of them are granule cells. They are relatively small. They don't really look like the very ramified neurons that you would expect to see in the cortex. And in the cerebral cortex, where we think a lot of the cool stuff is happening, there is a big imbalance in favor of non-neuronal cells. So, of course, that includes part of the epidermal cells and also part of the vasculature. But there's a fair number of these cells that are not these. And I usually show this really super old cartoon where you can see some of these non-neuronal cells. So there's a neuron here in yellow. We have an astrocyte making a bridge between the vasculature and synapses. And you can also distinguish microglia, the brain's immune cells, and oligodendrocytes, which are responsible for producing the myelin sheath. I guess in a more modern representation, you would also have maybe things like pericytes and intercapillary bridge cells that would be part of that picture. But this is really old. But we can do better than that. We can actually now do pretty cool reconstruction. So this is data from my friend Corrado Cali at the University of Torino, was acquired large blocks of EM data and reconstructed some of these cells. And I think for me, it really illustrates the complexity of the problem that we are facing, which is that while we know a lot about neurons, we don't know as much about these non-neuronal cell types. But also they form this really intricate mesh. And okay, we are diving now through a blood vessel. But yeah, you can appreciate that these cells are extremely ramified. Not only neurons are ramified, they connect in very complex patterns with neurons and with the vasculature. And they form this really, as I said, intricate mesh that is our brain. And I think for me, it illustrates the complexity we are facing. And I think it's probably impossible for us to understand how the brain works or how the brain gets sick if we are excluding these 80% of cells that are not neurons in the cortex or 50% of cells that are not neurons in the whole brain. And you will see, so this is about 100 microns, and this will now be broken down into some of its components. So there is a bunch of neurons in white that go to the top left corner. I think in the top right corner are microglia. Am I right? Yes. A few pericytes, a blood vessel, and some astrocytes. And these cells, these non-neuronal cells, they have some very exciting properties. This is a microglia that we recorded in London in rat hippocampal slices using a 2-photon microscope. On the left, you can see that this guy is basically extending and retracting processes all the time, touching its environment, contacting blood vessels, contacting neuronal cell bodies. There is evidence in the literature that when they contact neuron cell bodies, these neurons become less excitable. So there's a direct connection between a neuronal function and this mobile activity of microglia. And they also have a emergency response function, which you can see here. So if you blast a neuron using a laser, this is the meaning of this white circle that appears in the center, then they will rush there, extend processes, and rush to somehow circumvent the area that is damaged. I will talk more about that later. Astrocytes, of course, if you are familiar with the literature, they are less mobile than microglia. They are a little bit mobile, but not nearly as much. This is data from Baljeet Kak's lab in California. I mean, they did calcium imaging in mice in vivo, and you can appreciate the level of activity, of calcium activity that is happening in the cell body, but also all over the processes of these astrocytes. And so for me, I think it really illustrates again the complexity of what we are dealing with, and also the notion that these cells are mostly passive in the brain, I think is a complete fiction. And even oligodendrocytes that are rather more structural, you would think, have been shown 10 years ago to play a significant role in learning and plasticity. And this is an example from this paper, Mackenzie and colleagues in Science, and they did a very elegant experiments where they basically blocked the integration of, or the creation of new oligodendrocytes from oligodendrocytes precursor cells in adulthood. So these are animals that developed normally until adulthood, and then the integration of new oligodendrocytes was blocked by an injection of a drug. And you can see that they have to learn this rather difficult motor task because some of the bars in this spinning wheel are missing. So this is not super easy. So the guy on the left eventually manages to do it. The guy on the right as well, but he's nearly not as good. And that demonstrated that for this kind of task that involves fine motor coordination across hemispheres, you actually need this myelin plasticity also in adulthood. So this is not some kind of structure that develops during the developmental phase and that is then set for the rest of your life. So I think these cells have some very interesting properties. And so I'm going to tell you 2.5 stories about glial cells very briefly to try to convince you that really we should be looking at them a bit further. Okay, so the first one is about oligodendrocyte. This is work that I've done with my friend Kaylin Young, who is now at the University of Tasmania in Hobart. So if you look at a myelinated axon, you will recognize the basic structure of it. So you have internodes of myelin, rounds of myelin that basically insulate the axon as a cable. And there's, of course, the node of Ranvier between internodes. where you have ion channels that allow the action potential to be regenerated. And if you zoom even further, you will see that this is an incredibly complex structure, right? There's a very complex network of proteins and ion channels in the membrane, extracellular proteins that sort of connect the axon structurally and functionally, and also that connects the different the endpoint of the internode when the myelin essentially connects to the axon. So you might think that once this is built, you don't want to touch it too much. But in fact, stuff can flow here, so this is not completely sealed. And there's also this kind of empty space between the axon and the inner layer of the myelin sheath, which is called the periaxonal space. And from an electrophysiology point of view, if you wanted to really make the axon save as much energy, the myelinated axon save as much energy as possible, and if you wanted to maximize the propagation speed of the action potential, you would not want that. You would want the connection point to be completely sealed, and you would want to get rid of this periaxonal space. So my friend Kaylin, and in her lab, she basically stumbled on this accidentally, so she was involved in some other experiment, and that led to this story. So what they were doing is to do low-intensity repetitive transcranial magnetic stimulation using this intermittent theta burst stimulation, which is one of the things that is known to work for stimulating neurons using a magnet. And they eventually went and looked at the detailed morphology of myelinated axons. And what you see is that the large morphology of these mature oligodendrocytes does not change with ITBS, but we stumbled onto something, and something else is changing. So here is how the data looks like. So you do 2 weeks of stimulation, you sacrifice the animal, and you do some immunohistochemistry. You put that under the microscope, you can see that there doesn't seem to be much. And if you quantify, this is the length of internodes, and what you are going to see a lot of these graphs, this is basically all internodes, a cumulative plot of the distribution of their lengths. And this is a summary per animal to see that whether this actually holds when you look animal per animal. And we looked in the primary motor cortex and also in the corpus callosum, and we couldn't see really anything happening. So the distribution of internodes doesn't seem to be changing much. But what we did see is that this transcranial magnetic stimulation shortens the nodes of Ranvier. And you can see some data here, especially on the right there. So you have a visibly shorter node in the corpus callosum after ITBS than after the sham manipulation. And when you quantify that, you will find out that this is true both in the primary motor cortex and also in the corpus callosum. So earlier I realized that I messed up something with this slide, but essentially what they did, or what we did, was to do 2 weeks of stimulation, record data from some of the animals at that time point, and some other animals were also left to recover without any manipulation for an additional 7 days. And in the primary motor cortex, what we observed is that the nodes seem to be shorter after 14 days of stimulation, and eventually this disappears if you look at animals at 3 weeks time point, so after 1 week of recovery. But in the corpus callosum, you can see that after 14 days of stimulation plus 7 days of recovery, we still see a shortening of the node of Ranvier. We did verify that whether that was due to the integration of new oligodendrocytes into the network, or whether this is only happening in mature internodes. So using these transgenic animals where you can block the integration of new OPCs into the network, we repeated this experiment and essentially confirmed the same results both in the primary motor cortex and corpus callosum. So this is now different than what Mackenzie and colleagues observed 10 years ago. Their result was that you get plasticity, or some of the brain plasticity is enabled by active myelination that is probably activity-driven and task-driven in these mice that I was showing earlier. But here we are not talking about new oligodendrocytes. It's a restructuring of the microstructure or the microarchitecture of the oligodendrocytes that were already there in the network. The other thing that we saw is that this periaxonal space is also affected. So the periaxonal space is incredibly small, so it's in the nanometer range. So you have to use basically an electron microscope to really see any difference. And what we observed is that the G ratio was slightly affected in TBS. So the G ratio, for those of you who are not familiar, is just the ratio of the essentially the diameter of the axon divided by the diameter of the completely myelinated axon. And more interestingly, we saw an expansion of this periaxonal space. So essentially, you know, in sham animals, it's about 6 nanometers, while in TBS animals, it's about 9, 10 nanometers. So it expands a little bit. So let me just recapitulate very quickly. We were doing this ITBS stimulation. We looked at the motor cortex and the corpus callosum, and we see that the nodes of Ranvier get shorter and the periaxonal space gets larger. And eventually later in that story, we looked using a completely different protocol. So we looked into the fimbria, and we did a spatial learning protocol using this 8 arms maze. There's a food pellet at the end of each one of these arms, and the animal has to enter only once into each one of the arms. You can see a measurement of the errors that they are making that is progressively decreasing. So they eventually learn not to get again into an arm where they've already entered. In that case, we observed exactly somehow qualitatively the same results, but quantitatively the opposite. So the nodes get longer, not shorter, and the periaxonal space gets smaller. Okay, so what does it mean? Okay, so this is happening, as I said, in mature oligodendrocytes. It's reversible partially in some cases. And also there are a number of observations people have made that they have allocated to pathological changes that would be due to an overstimulation of the axon, for instance, or which you can induce in the lab, actually. But we didn't observe any of that. So it suggests that this is a bidirectional physiological mechanism that is task-dependent. So what is the effect of these structural changes? So my initial training was in computational neuroscience. So I basically went ahead. There's some biophysical models that have been designed back in the day, already in 2000, to simulate the propagation of action potential in myelinated axons. This is essentially, if you are familiar with the Hodgkin-Huxley mathematical model of action potential generation, it's sort of an extended, more complex model, but that is based on the same framework. There's actually a GitHub repository you can use where this has been implemented. And we also have modified extensively the model that was used initially by Atoll and colleagues. And so we have our own repository with the results of that paper, if you are interested. So there's a bunch of ion channels that we also kind of modernized or adapted from the literature. And so if you run this model on your computer, what you get is a prediction of the propagation of the action potential across this myelinated axon, and it reproduces some of the key features that people have observed when they've patched axons, like this depolarizing after potential, which I was initially surprised about because this is not what you usually see in these kind of models. But it seems that it is actually a biological feature. And then we basically took all the structural data that we had from the electron microscopy and fluorescence microscopy data from my colleagues, and we plugged that into this model to see what happens. So in the TBS scenario, what does happen? Well, very simply, so this is how it looks. So you can see on the left side, this is a recording of the simulated action potential at consecutive nodes along that axon. And we tried to play around with shortening the node of Ranvier, changing the structure of the myelinated axon, so expanding or retracting the periaxonal space, and also combining both things together. And what you see is that essentially what it does, as you would predict from biophysics, it essentially slows down slightly the propagation of the action potential. And the effects are summative. So if you combine both changes, the periaxonal space and the node of Ranvier, you get a reduction in speed that just linearly sums up. And we did that at 21 degrees and 37 degrees because eventually we'll compare that to some electrophysiology data in slices. But essentially, yeah, there's not much of a difference. At 37 degrees, everything is just faster, and the reduction is a bit more significant. So this is an interesting finding because basically it suggests that changing the size of the periaxonal space by a few nanometers, it doesn't seem like a very big thing to do, represents potentially an elegant way to modulate the conduction velocity. And if you run simulations with our model now, you can see that you can potentially change the conduction velocity between 1 and 4 meters per second. And we think it's interesting because if you predict the delay for the action potential to arrive at its destination on the presynaptic terminal over a distance of 1 centimeter, which would be relatively speaking, I think more or less the correct spatial scale for cross-callosal communication in a rodent brain, you can see that you can change the arrival time by a few milliseconds, and that would probably be sufficient to change the position where you sit in terms of causality. So if you are thinking about spike time independent plasticity, for instance, you know that there's this idea that you need action potentials to be fired in a causal fashion between the presynaptic and postsynaptic neuron, and that influences in which direction you have plasticity. So in that case, you know, these windows are a few milliseconds wide. So if you can adjust the arrival time by 6 milliseconds over a centimeter, that is already probably sufficient to change whether you have potentiation or depression. Yeah, and so okay, so I said in the Fimbria when we recorded after this learning protocol using the 8 arms maze, we observed the opposite structural changes. And the physiological effect is the opposite. So you just make the action potential go faster. So it's all very self-consistent. And so can we actually validate this? So my colleagues then did electrophysiology. They measured compound action potential propagating across the corpus callosum. This is for sham and TBS animals. And you can see, so if you vaguely remember the numbers I showed before, but we actually predict more or less the correct propagation speed in the sham case. And also the reduction of speeds that is observed in the TBS case is consistent with the model. And to my great surprise, because usually these things never work, with the same model, we were able to actually predict completely for free the change in velocity that was observed in the Fimbria, which again is self-consistent between the experiments and the model, and is also self-consistent with what you would expect from biophysics. Okay, so we are enthusiastically claiming that this is a new locus for plasticity, and that basically changing the size, the periaxonal space and the size of the node of Ranvier are places where the axomyelin complex has some capacity to adapt its structure that can change the arrival time of the action potential in a physiologically meaningful way. Just to conclude on that first story, as I said, I didn't say that, but underneath the myelin, there's a region here called the juxtaparanode, where that exhibits Kv1.1 and Kv1.2 channels. It's not entirely clear what their physiological role is, and we have a subsequent study that we are still busy writing. But to make a long story short, what it does is that it slows down the propagation of the action potential and essentially extends the dynamic range of this new form of plasticity. And we are also interested in looking and trying to figure out whether, because the shape of the action potential changes, whether that change in shape is sufficient to alter calcium entry in the presynaptic terminal, which could then change the release probability at the presynaptic terminal. So this is another story we are currently working on. Yeah, so these oligodendrocytes and the myelinated axon, maybe they are not as static as one as thought in the past. And you know, there's an interesting potential new type of plasticity using time delays that can be thought of. Okay, the next story I want to talk about very briefly is about microglia. So this is actually rather old work that I did when I was a postdoc in London. I didn't talk about that for a really long time, so I had to really refresh myself. So, you know, microglia were discovered by this guy, Pio del Rio Ortega, who was a student of Ramon y Cajal, and he published that. He published their structure for the first time in 1919, long ago. Yeah, more than 100 years ago. And there's a lot of them. So I think this is maybe something you underestimate. So this is a recording I did to photon microscopy in transgenic mice, where you have EGFP in microglia, and we are just diving from the surface of the cortex to about 400 micron or something like that. Actually, it's playing backwards. We are going up. Yeah, you can see the depths that is indicated there. So these are all microglia, basically paving the space in the rodent around the barrel cortex in these transgenic mice. And you can see some blood vessels on the surface at the end. So there's really a very large number of them. And as I said, one of their central properties is that they move. They move a lot more in developing animals, so in young mice or rats, and they are a bit more static as the animals get older. But for me, that was absolutely fascinating, because when you think about the structure of the brain, you're not necessarily thinking about a lot of tissue reorganization during adulthood, at least not at this kind of temporal scales. So this is over the course of 30 minutes or something like that. If you're recording relatively fast with the 2-photon microscope, you can actually see them moving by eye. You don't need to process the data. And the other mode of motility that has been well identified is that they will very quickly sprout many processes and converge to an area that you've basically blown up with the laser. And this was not a new discovery, so people had known that since at least 2004. And this is driven by ATP. So if you stick a pipette with a bit of, or quite a bit of ATP, you will see that you can reproduce exactly that mechanism. I have to speed up. Okay. And the mechanism that controls this convergence of processes to a damaged area is P2Y12. It's a purinergic receptor that is mostly sensitive to ADP. This had been identified by all of these people at various time points. And in fact, if you try to do one of these experiments in a P2Y12 knockout, they do eventually converge to the damaged site, but at least for the first hour or so, not much seems to be happening. While again, I mean, if you do this, it's within a couple of minutes you will see these processes converging. It's very, very fast, so it takes probably 1-2 minutes for something visible to happen. Okay, there's a different way, maybe a better way to quantify the propagation of the convergence of these processes. You can just draw a big circle around the area that you damage and just measure the propagation of the front of microglia. You can really easily quantify that. I'm just saying that as a token for later. So in there, we did some electrophysiology on microglia, and we identified a 2-podomain potassium channel that was expressed in these cells that seems to control their resting membrane potential. And while we were working on that, that story came up, and we could finally identify that the specific 2-podomain potassium channel that we were interested in is this 2-podomain allotane inhibited potassium channel, or TIC1. So what does this channel do? Well, it doesn't actually contribute to ATP-evoked motility, because if you block it using a drug that is somewhat sensitive to it, you will see that this process still occurs, and there is some quantification, right? So PSB is a drug that blocks this P2Y12 receptor. So if you block that receptor, essentially nothing happens. So you have microglia are not activated by this destruction of tissue. But if you block this potassium channel, nothing seems to be happening. So it doesn't play a role in that type of motility. But what we did observe is that it strongly affects, so this is now in knockout animals, but initially we did experiments using pharmacology. It does affect very significantly the microglia morphology. So you can see a wild-type microglia here from an adult mouse, and on the knockout animal has a very stunted morphology. And you can very easily quantify that using various types of quantifications. But essentially, however you look at it, it's very obvious from the small movie I was showing before that there is something very significant that happens if you block these channels or you get rid of them. And in fact, what we found out is that this channel is basically is responsible for maintaining this very ramified morphology, but also it plays a role in controlling the baseline motility of these cells. And again, you can see that in the knockout animal, these cells are much smaller, and also the kind of range of tissue that they scan around them, it's much more reduced, and they also do it at a much slower pace. Okay. All right. One thing that is interesting about this channel is that it's sensitive to gases anesthetics. Actually, that was the way we could pharmacologically verify that indeed that was the correct channel. So it's one of the, it's rather unique in that family of two-port domain potassium channels, because I think that most of the other channels in that family are actually potentiated by gaseous anesthetics. So by gaseous anesthetics, I should be more specific. I mean allotane, isoflurane, and sevoflurane. And so we thought that was quite interesting because, as you might know or not, if you get, or I mean, these anesthetics are not so much used for patients anymore these days. But in the 70s, 80s, you know, the allotane was used extensively in the US and in the UK, for instance, for big surgeries, either in elderly patients or in relatively young patients. And it is linked to severe cognitive issues. So in elderly patients, it can lead to all sorts of mental decline and dementia. It's a bit hit and miss. And in younger patients or kids, basically, it is linked to developmental disorders afterwards. So, you know, we are talking about several hours of anesthesia using some of these gaseous anesthetics. So we thought that was an interesting finding because we know now from this pharmacology that essentially whatever job microglia are doing while they are continuously scanning the brain tissue is severely inhibited if you're subjected to one of these gaseous anesthetics. And just to conclude that, and because I was told that there are immunologists in the room, we also, sort of a summary of the finding that we had. One of the central features essentially is efflux of potassium through these two-port domain potassium channels. In macrophages outside of the brain, people have sometimes related potassium efflux to the production of the NLRP3, or activation of the NLRP3 inflammasome. And in fact, that speculation in that case was correct. So when we've stimulated this microglia with LPS and ATP, so LPS is a component of bacterial wall, I think, so it's an activating agent for these immune cells. Essentially, you get a large release of interleukin-1 beta, which you can block using this TPA, which is this drug that blocks this channel. And we had supporting results in knockout animals where essentially the production of interleukin-1 beta by microglia in these knockout animals was very significantly reduced. Okay, so that's the second story on glial cells. And so I think, you know, it is just to point out again, there's a lot going on here which has not been as much investigated as neurons. And I think it is, to me at least, very obvious that we cannot hope to understand neuronal networks, function, and disease if we are not looking at these other components of the brain. And we have a follow-up story. I'm not going to, but we are basically busy digging further into the intracellular machinery that controls the mobility of these cells. And then just one quick mention about astrocytes. So I said earlier that there's a extensive, they display extensive and complex calcium activity, which I'm not quite sure we understand what this means. And there's actually one paper that I've been fascinated about for many years. I just never had time to work on it. It's the paper on the left here. It's this figure on the left. It's from Serrano and colleagues. It's from Richard Robitaille's lab in Montreal, where I think in hippocampal slices, they did a very elegant electrophysiological study where they delineated how astrocytes and local interneurons in concert regulate heterosynaptic depression when you stimulate Schaffer collaterals. And for me, it's a very interesting study because I think it's one of the few studies that really dives into the details of how astrocytes might contribute to synaptic plasticity in neural networks from a mechanistic point of view. And as I'm a physicist, I like mechanistic concepts and models. So we've recently built a relatively large model of the hippocampus. You can see a simulation running here. This is work from a Ukrainian student of mine who is a very gifted programmer. And we have not yet introduced astrocytes into these simulations, but we have validated as much as we could the behavior of this simulated hippocampus, or slice of, or tranche of hippocampus. And it reproduces a lot of the electrophysiological behavior that people have observed over the years or sometimes speculated about. And so we are really excited about it, and we are just finalizing the paper now, but we really want to go and try to look at what astrocytes are doing in that context, potentially modulating the activity of this network. Okay, so that was a lot. All right, so now, I don't have a conclusion slide, sorry. That's the end of the science I'm doing. But yeah, I hope if you were not interested by glial cells before, I hope I triggered you into looking more deeply into their role. And if you knew already all about them, I hope at least you found these results potentially interesting. Now, so when I was invited here, I agreed to talk about the work I do in science policy, which I've been doing for about 10 years. And so maybe I will run you through, how did I get into science policy and why, and what do I do now, whether this is useful or important, which is an open question, and whether it was worth it, maybe from a career perspective. So how did I get into science policy? So this has been my career so far. So I entered university in 1996. I did essentially bachelor and master that was in Lausanne in Switzerland, then a PhD at EPFL. I had then a long, painful period that most of you will be familiar with 3 different postdocs in Switzerland and the UK, and some research stays in Japan. Eventually, I landed something like an assistant professor position in Geneva, and in 2021, I moved to Maastricht, where I'm now a full professor. So that's been my academic career. I think a very typical career for someone of my generation in neuroscience. So I trained as a computational neuroscientist, and then became an experimental neuroscientist transiently for a while, and then I'm back to being a computational neuroscientist. And so my science policy career essentially starts in 2014 when I joined the Marie Curie Alumni Association. So I had the Marie Curie Fellowship with which I could go for 2 years as a postdoc to the UK. It's an excellent scheme. I highly recommend it if you want to move abroad. It's a relatively high salary for a postdoc for 2 years. And, 2 years is short, but it's better than a lot of other fellowships. And it's relatively achievable if you have a decent CV outside of your PhD. So I wasn't really very active in science policy until then. And then I was elected to the board of this association in 2018, then to the board, or was dragged into the board of the Initiative for Science in Europe in 2019. And since 2022, I'm the stakeholder representative at the European Research Area Forum at the European Commission, and I will explain a bit what that is. Okay. So why did I do it? So towards the end of my postdoctoral years, so I'm a first generation academic. My parents didn't go to university. And towards the end of my postdoctoral years, I got to the conclusion that if you want to be successful in academia, there are a lot of unwritten rules. There's a lot of, your scientific productivity is not the only determinant of whether you're going to be successful or not. And if you don't have any kind of mentoring or support network around you, people who can tell you how the system works, what hiring committees care about, this can be relatively tricky to figure things out on your own. And so as I was getting towards getting a first faculty position, I thought that if I ever had the chance to have my own lab, I would dedicate some of my time to mentoring early career researchers, because I wished I had benefited from such mentoring, and I still do it. So I'm actually mentoring 2 or 3 younger researchers at the moment, and so that was my personal motivation to do that. But then, as I was relatively active in this association, the Marie Curie Alumni Association, I discovered a much broader world than I had signed up for. And also I was progressively, because I don't know how to say no, I was progressively dragged into increasing or more and more different roles. And so I didn't do anything to get there really very proactively. It almost happened all by accident. And often I was put into this role because I had made friends who then asked me to serve into these consecutive boards. And that was the case for the role that I'm doing now. So what do I do now? So I sit in this ERA forum, ERA forum, and I'm a stakeholder representative for individual researchers and innovators. So I think that, in fact, I represent all of you, plus everyone else who vaguely fits that category in Europe. And what is this forum? So this is from the Austrian portal. Essentially, it's a forum that is organized, it's an expert group that is organized by the European Commission. Are sitting in that meeting or in these meetings representative of the member states, so usually people who work at the Ministry of Education and Research of their respective countries. Some other countries that are invited. And there's also a number of stakeholder representatives. So myself, but there's also a bunch of other people representing universities, funding bodies, research infrastructures, and so on. There's actually a very large landscape of organization and people who are involved in that. Too many, in my opinion. Is this recorded, this talk? Can I say stupid things? It is, okay. So the main tasks of the forum is to essentially co-design and coordinate this policy agenda, which is essentially a list of actions and key points that the European Commission and the member states are trying to work on. And also area actions. So this is actually the detailed content. There's more stuff in there. So this sounds very theoretical, but in fact, you will see that it has quite an influence on potentially all of us. So this is the content of the current policy cycle. I don't know if you can read, but it covers a very broad, a very long list of different activities, right? So you have equity in open science, research infrastructures, inclusive intersectional gender equality, making research careers more attractive and sustainable. The typo is not from me, it's from the Commission. And this is actually directly targeted at the early career researchers. Research assessment, it's also targeted at the early career researchers. Certain things that are meant to make Europe more competitive. The global approach to research and innovation, which is basically where the Commission and the member states are dealing with how we interface scientifically with all the other scientific powers, like the United States, China, and so on. Yeah, so there's a lot of things in there. It's very, very broad. And the ERA forum is actually the forum where all of these things are sort of coordinated. If you want to know all about it, there's actually a website that is new-ish, and that is not too bad. So it sort of contains all of the information. So the concept of the European research area, it exists since the early 2000s, but this ERA forum in this new format is only 3.5 years old. And this was the first cycle of 3 years from 2022 to 2020. End of 2024, and now we are in the second cycle. And if you want to find information about it, you can look at the website. There are some reports that, for instance, characterize the performance in research of Europe, which sadly you will see is not great, has been stagnating or flagging since the financial crisis of 2008. So is this work useful or important? I don't know, because to give you a sense of how it works, there is no mechanism for taking decisions, so it's always rather unclear who is making decisions or why. But at least we are sitting at the table and we have a voice, and so we can argue for the things that we think are important. Unfortunately, at the European Commission, there is kind of a bureaucratic streak. They like to control things top down. They can see that Europe is not doing fantastically well economically or in R&D in comparison to other great scientific powers. And they think that they know better how to solve that issue, which they want to impose from the top. So again, this might seem very theoretical, but this will translate often very quickly into directly into your career. For instance, there's a discussion at the moment, the Marie Skłodowska-Curie Action, which is a really good postdoctoral program for you to go 2 years abroad. At the moment, you can just apply to do anything you want. So it's really bottom up. But the Commission thinks that there is too much of these resources are being spent on useless research. So they want to make that a lot more top down. They want to basically give pointers as to what kind of research you should be doing and what kind of projects you should be submitting. So that was one of the few programs that the European Commission had that was really where you're free to propose the research that you think is the most interesting or the most creative, and it might be taken away very soon. The same discussions are going on about the European Research Council, which is a more advanced, what you apply for at a more advanced stage of your career. So I think it's important that we are sitting there and that we are pushing back against some of the idiotic tendencies that some of the people at the Commission have. They are lovely colleagues, but one thing you need to understand is that the people who make these decisions often have no scientific, technical, or engineering background. They typically have a background in law or European affairs. No offense to, I hope no one here has this kind of background, but they will typically have a bachelor in European affairs and then went into government, and they could be managing this or pensions that wouldn't make too much of a difference. So they mean well, but I think they don't always fully understand what they are dealing with and maybe the specific peculiarities of the research world. And they have no field expertise, right? They have never worked in a lab, they've never published a paper, they've never started a company. And I think this is really an issue. If you think about the great branches or entities of the state, you know, you entrust the military to military men and you let the healthcare system being run by doctors. But for some reason, scientists are being run by people who are not scientists, which I think really is a problem. Okay, so this is what I do at the moment. Was it worth it? This, again, I don't know, because we are voicing our concerns, and sometimes we win, sometimes we lose. It's not always clear. It definitely, for me, had an impact on my career, because I was elected to the board of this association. I had never thought about being elected to anything. But then when I found out that it's actually quite easy, I got elected to a lot of things, right? So I got elected to the board of an organization in computational neuroscience. I got elected as a chair of the Science and Technology Council of EBRAINS. And then I applied for this Foresight Institute. I was asked to be a polymath fellow at the GCSP in Geneva. And I'm chairing the ANR committee in France, and also, yeah, I was just asked by a friend to join this thing. And this really had a positive impact in my academic career, not, I think, because I deserve it, I don't believe that, but because I've been very active, I built a huge network, and also being elected to the board of this association was kind of a sandbox. We could do stupid things, it didn't have too many consequences, and it gave me, I guess, the confidence to apply for bigger roles into my academic careers. And so if you're afraid of being in charge of things, I would highly recommend you know, being active in that regard. And then in return, I got several awards. Yeah, and again, I'm not quite sure I deserve these awards. There's something funny that maybe you don't know. You don't get awards, you ask for them. You basically ask your friends to nominate you for these awards, and this is how it works, okay? So I've been very shamelessly asking friends to nominate me, and they were kind enough to then give me these awards. And so I think, again, you know, you don't get a Nobel Prize right away. Not, I will never get one, but you know, you work your way towards bigger and bigger awards. And so if you are thinking about doing an academic career, this might be a useful way to, you know, beef up your profile. And unfortunately, maybe it matters to get this kind of recognition and awards, whether they are deserved or not, because this is for the ESC Advanced Grants 2025 call. You can see that one of the criteria to judge whether your project is a good project or whether your profile is suitable for the ERC is peer recognition, right? Provide a list of selected examples of significant recognition by your peers, such as prizes, awards, fellowships, elected academy memberships, invited presentations, and so on. So whether this is a good direction for academia to validate this kind of behavior and this kind of things, I think is an open question. But you know, you might want to consider these kind of things. So when you're going to conferences, you know, try to don't overlook best poster awards, travel grants, and these kind of things. You know, they will be important in building a profile towards bigger roles and bigger awards. Okay, so with this, I will leave you on these words of wisdom. So make friends, keep them, and try stuff. There's a lot of opportunities out there. It's not always clear which ones are good and which ones are not good. But you know, you should really free yourself from any kind of fear of trying things. And you know, I leave you with the words of this great philosopher Bruce Lee: "Be like water, yeah, flow around the obstacles." That's it. Thank you for your attention.
Thank you so much. This was extremely captivating for me, definitely. I hope the audience shares my view on this, both parts. scientific and your policy work is fantastic. I will allow now to open the discussion. Okay, I see the very strong hand already shoot up to the sky. So here I'm running.
Thank you, Mateusz. Thank you, Renaud, for this fantastic talk. I have a specific question regarding the last part of your presentation. I wanted to ask about the policy for animal use in research. Can you comment on where we are going, and should we switch the labs, shut off the labs which are working with animals, or do you see hope that this will be still possible?
No, so I think, okay, so we are not going in a good direction. That's my opinion. There is a significant push, and we were just discussing that at lunch, actually. There is a significant push at the European Commission so there is one of these actions that is in the list, which is called as a very incomprehensible names, and it's clearly on purpose. So what is it? Accelerating new approach methodologies to advance biomedical research and testing for medicinal products and medical devices. And this is clearly a Trojan horse to ban animal experiments, right? It is not worded like this because I think the people who've pushed for this action, they know very well that there is a lot of pushback from the scientific community and people who are involved in animal experiments. But it is actually in one of the earlier meetings when that was discussed, there was a clear statement from the representative from the European Commission who said, we want to phase out animal experiments in Europe. Yeah. And so I think we really need to push back against this because this was proposed by the Dutch government, unfortunately, and also by some entities within the European Commission that are actually not involved in regulating or organizing animal experiments. They, I think, are more in the management of ecosystem. So for me, it's very mysterious why these people all of a sudden care about this, why they are being listened to. The Dutch government, I think, has made a strategic mistake in making a commitment to the population to actually push for this kind of actions. So there was a lot of pushback from some of the member states, but unfortunately, the action went ahead. And I think one of the things I've learned from this is really that we are very disorganized as scientists. And the decisions are left to people who are very emotional about it, who have actually very little understanding about biomedical research or the constraints that we are already under when doing animal experiments. And I think we really need to make an effort to be better organized. And I can give you a concrete example. While this was being discussed at the European Commission, we tried to coordinate among ourselves to push back against this within the frame of this forum. So I made several interventions. I was labeled an angry scientist and an undiplomatic. But the animal rights groups wrote countless letters to all directly to the ministries, or directly to the people, the political level who are sitting in power. And so they are, of course, under quite a lot of pressure. And I think that in comparison to these animal rights groups, we are far less organized and far less efficient in communicating. And it's annoying because you would like to focus on your research rather than doing this. But unfortunately, if we don't do anything about it, this is going to be it will become increasingly difficult to do these kind of experiments. And then maybe one day completely banned. Yeah. And they think we can, I'm a computational biologist by training. These people think we can simulate everything, which I think is a complete fiction. And the problem is that that's the tip of the iceberg in a sense, because a lot of these actions, behind the rather obtuse names, also have a direct impact on your research and on your capacity, sometimes less so than the one on animal experiments. But it's the same thing, if we don't get involved, things will be decided for us. And rarely it will be decided in a direction that is good for us, I think, or good for science and good for European competitiveness.
Thank you.
It's close.
Okay, very good talk. I would have a question about the first part of about the oligodendrocytes. Have you been observing, for example, the shortening of the myelin, but nearby the synapse? Because you have been showing that it is shortened or length and more longer after the TBI. But have you been observing, or is it even possible to observe the changes about the myelin around the presynaptic part of the neuron? Because then it would be directly also be capable of changing the electrochemical capacity of the calcium entry into the presynaptic nerve synapse.
Sorry, there's a lot of echo. I'm not sure I got your question correctly.
Have you been observing the changes of myelin, like the width or the shortening or lengthening of the myelin around the presynaptic terminal of the synapse?
No, because it's very
TBI?
it's very difficult to do, because the periaxonal space you can only really see in electron microscopy. And then you would have to track, an axon over until you get to the presynaptic terminal, and for the rest it's immunofluorescence. So these structures are so small that and also over, well, they are small. The changes are very small, but the structures are also over extended lengths, across, for instance, the corpus callosum. So it makes studying this rather difficult. You can't hope to manipulate it, in a slice or something like that and see a change. At least I'm not quite sure how you would do that. And that's why we have, for instance, we have like hundreds of nodes. We collected lots and lots of data because we had to get to an answer from looking a bit randomly in the tissue effectively, right? Because you cannot track, for instance, what happens along a continuous axon. There's actually a paper from my former boss where they tried to do that. But they had one data set from a Japanese group where they had tracked 3 separate axons over a few nodes. And I think it's one of the few data sets where people have, at least at that time, they could see a few consecutive nodes. But it's like 5 or 6 nodes over 3 axons, or 3 separate axons, and there's a few nodes for each one. So it's pretty, unfortunately, this is pretty limiting. Maybe with expansion microscopy, where you can image the whole brain, this is going to change. So actually, I have a collaboration with a friend of mine who is at Pasteur, and he has built ExaSPIM, and so he's already started collecting data from a first expanded brain. And we hope to set up a collaboration with my friend in Tasmania to see if, for instance, you could track, axons across the whole corpus callosum and see what we can see there. But we don't have any data yet.
Okay, and the second question would be probably not that scientific. You have shown the question about, is it worth to go for this policy of the scientific policy career, and you have showed the achievements as the goals or something which could be accomplished by having this career. But I would have more rather the Marcusean critique. Are you happy with this career?
Well, no, it's been yeah, because I think it's something that matters and it's something that matters for your scientific career or our scientific careers, that we have an environment that is suitable for doing good science. Yeah. And unfortunately, it's not entirely down to us. I mean, there's a big machine around you. And, as you grow in your career, I think it's also your responsibility to understand the professional environment in which you evolve. And I can tell you, from the transition from being a postdoc to being a group leader, it comes at you whether you want it or not, right? I mean, you will be involved in a lot of decision making in your institution, and in turn, these institutions are influenced by their local government, the region, sometimes the city, or national government, or the European Commission. And so it's at least useful to understand what's the process behind these kind of decisions, I think. Am I going to do that for another 10 years? This I'm less sure, because I've sort of seen it by now, but, and I've done a lot, but, yeah, I think it was important, and it was not a revelation, but at least it expanded my horizon quite a bit, and it was useful in that regard.
Last question. And then after this, there will be a meeting with young scientists.
Thanks for the talk. It was a great talk. I have actually a couple of follow-up questions from the two first. I'll start with the last one. You told us that in those meetings, in these forums, you have a lot of people that have your background, so our background, scientific background, but then you have a lot of people that don't have. Objectively speaking, do you think having the people with the scientific background moves the needle at all? And does it move just slightly on the decision part?
Yeah, I think so. I mean, it's again, you know, it's difficult to really know for certain because the decision process is rather obscure. And, you know, in these meetings, you have like, I would say 200-300 people are sitting there. Some are observers. But I think you can at least voice the concerns of scientists, of people who are doing things, who actually have, you know, boots on the ground. And if you're not there, you can't. I think on balance, it's probably better to be there, if only to know what's coming. You know, there's also clearly a information role. And I can see, for instance, since I've been sitting in these meetings, I hear something in these meetings, and 6 months to a year and a half later, I'm sitting in a faculty meeting and we have the same conversation, right? So I can see how it trickles down from me sitting in Brussels to the faculty meetings in Maastricht within 6 months to a year. And all of a sudden, you know, and everyone at the faculty meeting is complaining, yeah, who decided that? Why do we have to do this now? But you can see the process. And I hope have a chance for certain things to be stopped or altered on certain occasions. And I think it would be dangerous not to have any kind of representation. Now they are people who do that professionally. So the university associations, you know, they have people dedicated to essentially lobbyists who are based in Brussels. But again, these people are often quite disconnected from the reality in your institution or in your laboratory. And I think it's important to bring the voice of people who are actually doing the work. Yeah, because they are talking about sustainable careers. But, you know, these guys have been working in a ministry for 20 years. What do they know about moving every year to do a different postdoc and so on? They just have no, I don't want to sound too negative about them. I think they just have no personal understanding of what it means, you know. And so it's important that you can be there and say, hey, you know, look, this is what you have to do in an academic career. These are the constraints. This is the cost on your mental health or your, you know, personal life and things like that. And so if you are not there, you know, they will just decide for them for what they think is a good decision, and you have no input on that, which I think would be very risky.
So the second one, regarding the first question about animal experimentation, do you see in those meetings any prior insight that will tell you how they would try to enforce that? Because banning the animal experimentation would take a long time for them to do. So I'm assuming the first way that they would approach it is through pressure on the funding body, and they would probably stop funding research that has a lot of animal experimentation. Do you think there will be like a loophole in which they would still fund experimentation if it's not done in Europe and in collaborations?
I don't know. So the push that they are making now for this new so-called new approach methodologies, you can see that they are building on the notion that you can do biomedical research like you produce nuclear bombs by just simulating reactions. And that's enough for clinical trials, for basic research, for toxicology screenings, and so on. And so what they are doing is, so they started pushing this like maybe 2 years ago, and already in the framework, in the framework program, in the current framework program, there were grants that were given to, you know, synchronizing entities across Europe on these new approach methodologies, and also developing standards and developing new approach methodologies. So my suspicion is that they know, realizing that banning animal experiments outright is too much of an ask today. They are trying to push the other side to say basically, well, we can, you know, spend a lot of money on the people who are moving away from animal experiments and who just are simulating everything. So it's great for me as a computational biologist. And that eventually, you know, they will kind of strangle animal experiments that way by moving a lot of funding away from this. Then by asking that if you do animal experiments, you have to have a component on new approach methodologies and so on and so on. And it's clearly, I mean, it was very hypocritical from the people of the Commission, in my opinion, because the guy said in a meeting outright, we want to phase out animal experiments. And then as there was a pushback, you could see that they changed the language progressively to make it more and more acceptable, so to speak. And unfortunately, a lot of the member states committed to this action, which was because they saw the language had been softened up enough to be acceptable. But behind that, there's clearly a desire to get completely rid of animal experiments. Yeah.
Great. Thank you again for a wonderful talk.
Thank you.
Big applause.